This procedure will demonstrate how to prepare an undercut model of posttraumatic epileptogenesis in rats. The first step is to make a simple device that improves the precision and consistency of the undercut surgery. Then the animal is prepared for surgery, and a cranial window is cut on the surface of the skull to provide access to the brain. Finally, the needle of the undercut device is inserted into the white matter of the cerebral cortex and rotated to produce a fan-shaped undercut in the target region. Ultimately, results can be obtained that show epileptiform activity in brain slices through field potential recording.
The main advantage of this technique is that more precise and consistent cortical lesion can be made with a simple, custom-made surgical device.
This animal model can be used to answer key questions related to epileptogenesis after traumatic brain injury.
Though this method is often used in rats, it can also be applied to mice.
Generally, people new to this method will struggle because making such precise lesion of rodent cortex with free hand is difficult.
The surgical device consists of 3 parts: a supporting plate, a guiding tube, and a needle bent at 90 degrees close to the tip. To prepare an undercut device for surgery in 3 to 4 week old rats, first cut a piece of 1 to 1.5 mm thick 7 to 10 by 30 mm rectangular stainless steel or transparent plastic to make the supporting plate. Take a 25 gauge and a 22 gauge